part 4 for the chapter 23 on lipids. This is really a summary. I'm not going to ask you too many detailed questions. Just read your textbook and the summary about it. And I really will ask you basic questions about the different type of lipids we can find. So first, hydrolysis of triacylglycerol. Like all ester, they can be hydrolyzed. In the body, this is going to be catalyzed by an enzyme, hydrolase, and this is the first reaction in the digestion of dietary fats and oil. Another thing I would like you to look a little bit more carefully is soap. Well, the soap is a mixture of salt of fatty acid formed by saponification of animal fat. And here you'll see the structure of a micelle. This is a spherical cluster formed by the aggregation of soap or detergent molecules so that their hydrophobic ends, hydrophobic, they don't like the water, are in center and the hydrophilic ends are on the surface. This is here the representation. If you look at the micelle, the hydrophobic is inside where you have the fat, the hydrophilic is going to be on the outside. So the way soaps work as cleaning agent is because they have those two ends. One is hydrophilic, meaning that it likes the water, and is going to be able to dissolve with the water. The long hydrocarbon chain portion of the molecule inside, the water-fearing, tends to dissolve the grease. Once you have that suspended with micelle, the grease and dirt can be rinsed away. That's the way soap works. Three major kinds of cell membrane lipids in animals are phospholipid, glycolipid, and cholesterol. So what are cell membrane? Well, they are the ones that separate the aqueous, so water-like interior of cells, from the aqueous, water-like environment surrounding the cells. Membrane establish a hydrophobic, repel the water barrier between the two watery environments. And the lipids are perfect for this function. So in other words, that's the way the cell membrane is going to decide what can get in and what can get out. So among those, I would like you to review phospholipids, lipid that has an ester link between phosphoric acid and alcohol. Phospholipids are similar to soap and detergent molecule in having ionic, hydrophilic heads and hydrophobic tails. However, they have two tails instead of one, so this is what we call the phospholayer, the bilayer of phospholipids. They are going to have their double two tails facing each other, and then the hydrophilic head pointing to the outside and the inside of the cell. Glycerophospholipid or phosphoglyceride, well, those are the most abundant membrane lipids. Example here, sphingomyelins are the major component of the coating around nerve fibers. Glycolipids, well, they are also here derived from sphingosine. They have a carbohydrate group instead of the phosphate bonded to an amino alcohol. They reside in cell membrane with their carbohydrate segment extending into the fluid surrounding the cells. That's an example here on the right. Then we look at sterols. They are soluble in hydrophobic solvents and not in the water, so they are classified as lipids. So lipids, and the characteristic is the fact that you have a four-ring carbon skeleton, so a tetracyclic carbon skeleton. This is here just the steroid nucleus, typical. Among them, cholesterol, most abundant animal sterol, very important for cell membrane and starting material for all other sterols. Within the cell membrane, nearly flat cholesterol molecules are distributed among the tails of phospholipid, and that's what gives you that mosaic, fluidish shape of the cell membrane we're going to study later. Cholesterol, well, I'm not going to ask you to remember the molecule. Just however, you should know they are more rigid and hydrophobic than the hydrophobic tails. Cholesterol molecules help to maintain the structural rigidity of the membrane, and about 25% of liver cell membrane are made of cholesterol. Bile acids, well, they are very important to be able to emulsify fats during the digestion. They are synthesized in the liver cells from cholesterol and they are stored in the gallbladder. Those have a polar end and a non-polar end. And finally, just take a closer look at steroid hormones. 3 types: mineralocorticoid, such as aldosterone, very important for the balance between sodium and potassium ions; steroid hormones such as glucocorticoid, to help to regulate glucose metabolism as well as inflammation. Most anti-inflammatory ointments contain hydrocortisone to reduce swelling and itching, which are some examples. And of course, the sex hormone. The two most important are androgen and testosterone, as well as androsterone. Estrone and estradiol, the estrogen, are synthesized from testosterone. I like to show you that picture because sometimes students tell me, oh, I just forget a double bond, or I just forget a OH. Well, if you look at here the difference between testosterone on the left and estradiol on the right, you see that they look very similar, and yet they make all the difference between men and women.